We have already ascertained that DNA is the genetic molecule, contains the genetic information. Now people wanted to know how a DNA molecule makes another copy, identical copy of itself. There are three models we can look at, people proposed. There is the semi-conservative model in which two strands separate, a new strand is formed with the older strand. So it at the end result would be you will have two hybrid strands, one each containing an older or parental DNA molecule and a new DNA molecule, as we can see in the option A on the slide. The conservative model is the two DNA molecule strands separate, new strands are formed, and somehow the original parental strands again come together, and the new strands which have been formed, they also come together and form that hydrogen bonding between the bases. There's dispersive mechanism of replication in which some parts of old and some parts of new DNA molecule are present in the progeny DNA molecule. So there are three models. We will look at a very elegant experiment how scientists were able to discern which of these models is correct, which of these models is actually the model which cells use to replicate their DNA. Let's look at this experiment, Madison and Salthal experiment. Here again we have bacteria. Bacteria need nitrogen atom. Nitrogen is, as you know, important component of amino acids which make proteins. So these scientists grew bacteria in heavier isotope of nitrogen, an isotope of nitrogen in form of ammonium chloride. That was the source of nitrogen. So when the bacteria were growing in heavier isotope, the nitrogen was incorporated. We are talking about DNA, not the amino acids. Nitrogen is incorporated in the bases. Nitrogen atom is present in the bases of DNA. So when these bacteria were allowed to grow, after certain amount of time, when they had successively replicated, these bacteria have multiplied for several generations, their DNA was extracted and was put in the centrifuge. Centrifuge, we have already seen how we use centrifuge. Centrifuge basically increases the gravitational pull of the sample, gravitational pull on the sample. So whatever is heavy will come to the bottom, whatever is light will stay at a higher level. Here you can see after growing the bacteria in heavier isotope of nitrogen, when they extracted the DNA, the heavier DNA, because of the heavier nitrogen isotope, made a band at a specific or a lower location in the test tube which was put in the centrifuge. After these bacteria had incorporated heavier isotope of nitrogen, they were shifted to a normal growing media where the normal isotope of nitrogen present in the ammonium chloride, which is N14. So with this, when they extracted the DNA from this sample of bacteria, they had allowed bacteria one round of replication in media containing normal nitrogen. The band shifted slightly upwards. So this basically negates the conservative model of DNA replication. It could not have been conservative. Had it been conservative, we would have seen two bands here, one at the original location. Where did this band go if it was a conservative method of DNA replication? So this part of the experiment basically negates the conservative method of replication. However, it does not negate the dispersive mechanism of DNA replication. In the next round, they allowed the bacteria to grow again in the normal media containing normal isotope of nitrogen, N14. Now they had two bands. They had one band here at the intermediate location and they had a light band. If the model of DNA replication was dispersive, they would not have been able to get this light band right here of N14. Because if it was dispersive, the band would have appeared somewhere in between here. That did not happen. This conclusively pretty much proved that DNA, the model of DNA replication, is semi-conservative, not dispersive or not conservative. So let's look at those three models, A, B, and C. So A model is the correct one. This semi-conservative mechanism of DNA replication is the correct one. I have put a check mark there. The other two mechanisms are not what happens in nature. We'll continue our discussion of DNA in the next module.